Hello and welcome to the EMJ Podcast with me, your host, Dr. Jonathan Sackier. Today, I'm joined by Dr. Jose Alexander. He's a clinical microbiologist and director of microbiology at AdventHealth in Orlando, Florida. He was born and raised in Venezuela, and Dr. Alexander earned his medical degree from the Central University of that country before moving to the United States. He's now a leader in antimicrobial resistance, something I find absolutely fascinating, epidemiology and advanced molecular diagnostics. So we have got a lot of interesting things to discuss. He pioneered a rapid test for a specific organism that we are going to hear about. Pretty fascinating stuff that reduced diagnostic time to 3 hours. Very, very critical given how nasty this particular infection is. And his work in next generation and whole genome sequencing is transforming the detection of tuberculosis and pneumococcal infections. Beyond his medical career, the good doctor likes working on cars and restoring old classics. And after a long day, he unwinds by playing bass. And I'm a huge music fan, so I'm looking forward to hearing about that. So welcome to the EMJ podcast, Dr. Jose Alexander. Real pleasure and honor to have you.
Thank you very much. I really appreciate the opportunity. Thank you.
You're welcome. So I always like to learn about what makes people tick and, non-medical stuff. I used to love playing with cars back when I had a life. So if you had a dream car to play with, what would that be? A, and B, what kind of music do you like playing?
Well, I think I actually worked in my dream car for a while. Sadly, I don't have that car anymore. There was a Volvo P1800, a classic
Oh,
GT from the 70s. An
absolutely.
amazing, beautiful car.
It was, if I recall correctly, it was the mode of conveyance for a chap named Simon Templar in a TV show called The Saint.
The Saint. Yes.
Yeah, beautiful looking
Yes.
machine. Oh,
Yes.
wonderful.
Yeah.
And
Well,
well,
I think one of the interesting thing is that a dream car will be something like a DB5, right? A DB4 Aston Martin.
well,
But
that's my dream as well, my friend. That is my dream car as well. Beautiful
but
machine.
in the lines of those GT from the 60s and 70s, I think the P1800 is a pretty good looking car.
And lovely on the bonnet. It had in chrome Superleggera.
Super Leggera,
Touring
yes.
of Milan, beautiful. I've driven a couple of them, and they're just amazing, beautiful motor cars. Okay, so what kind of music do you play?
So I learned to play bass during the pandemic. So when I was not at work, I just had to be at home. I just started playing bass, and I actually love jazz, blues, and a little bit of like 60s, 70s classic rock. I like those. Just to play over those tracks and just to forget about everything else for a few hours.
Well, if we lived closer together, I'm sure we would be having a beer and we'd have a lot to talk about. I
That to
was
talk
out
about.
last night watching great musician Madeleine Peru, and her bass player was absolutely awesome.
Amazing.
Anyway, I suppose we ought to talk about microbiology. What got you into that and your specific areas of interest?
So when I actually went to my medical school, I already have an interest for infection disease. I remember when I was in high school, we have me and one of my classmates, we had to present about some topic in biology, and I have this old Discovery magazine that was talking about phage therapy, the Eliabat Institute. And that was in high school. So when I went to medical school, microbiology was probably one of my favorite topics, and I decided when I finished, I want to dedicate my time to that. So I trained as a medical microbiologist, but specifically antimicrobial resistance. Since that moment, I already got really into the perspective of knowing the organism for having a better perspective of how to treat patients. I think the clinical knowledge has been the foundation of my career, but being able to know the organism to a very detailed level is the another component that I think that me putting outside of the clinical side, but just in the microbiology, being able to convey that type of information to clinicians I think I can provide more insight for them to be able to treat the patients in a different way. So I think that's it's kind of like a little bit like an intelligence into the world of the organism to take advantage of how to treat
Yeah, it's I was always fascinated by it at medical school and trying to. The thing that always got me was the conversations about whether a virus met the criteria to be considered a living thing
them, living organism.
and an organism. Just absolutely fascinating. Well, so there's
Yeah.
one particular, I just can't wait to ask you about this because I remember the first time I read about it, a free living parasite.
Mhm.
And I'm probably going to slaughter the pronunciation of the name, Nagliera fowleri.
Mhm.
It's an amoeba that has a taste for the brain. And when I heard about it, my desire to jump into lakes or swimming pools was rather quelled. It apparently kills more than 97% of people who it infects due to delays in diagnosis. You developed a test, as I said in my introduction. Walk us through the process of developing that test for this thankfully rare but very, very nasty critter. And tell us about the impact that's had on patient outcomes, please, Jose.
Sure. So we in AdventHealth we have the experience to encounter two patients with this infection. Sadly, the first patient expired. The second patient was the diagnosis was done on time. So we put a lot of emphasis on the diagnosis. So we see the three pillars for this type of disease: right? Diagnostic, a suspicious diagnostic, then diagnostic confirmation and treatment. The diagnostic pillar has always been the weakest. Education informed physicians to suspect this type of infection based on epidemiological criteria. But the diagnostic was done at the CDC, so sample has to travel there a few days and then you get the result back. And this disease actually can kill the patient in just a matter of days, a week. So for us, the diagnostic was that major barrier that we needed to break through. We used to have some microscopy-based diagnostic method, but there was always the operator-dependent variable that we need to make sure to remove because if you don't have experience looking at a microscope and looking for a parasite in the CSF, that is a really tricky condition that not everybody have the experience to be able to do. So we decided that at the time we needed a molecular diagnostic, something that is unbiased, something that is operator independent. So during the pandemic, during the COVID pandemic, we had a lot of molecular diagnostic platforms and assays and reagents that was left behind. So we start working with those different components and start building a rapid PCR method. So we develop a PCR that is able to identify free living amoeba, Nagliera fowleri, Balamuthia, and the other one is Acanthamoeba. So the idea was that when we have these strong suspicions and we have access to the CSF, we don't have to send a sample to the CDC. We can actually run it in-house with a high positive and negative predictive value. We can provide that feedback to clinicians because the diagnostic at this case ruling out the presence of this infection allow the clinician to focus the diagnostic and treatment in other infection disease organisms, right? We also run viral testing in them. But the treatment can be really aggressive for this disease. So a false positive can also have consequences for the patient. And a true positive will put the patient in a rapid treatment. So that is critical for the survival of this infection. And I think that was the goal that we tried to accomplish. We've been doing this for 2 years. We have multiple potential cases that we were able to rule out on time, 3-4 hours. So the clinician now feel more comfortable and confident that they have evidence that there is no free living amoeba eating the brain, so they can focus the diagnostic to another aspect. So this test is open not only for AdventHealth. We open this test to any patient in the U.S. or even internationally. That was
Just for my edification, tell us a little bit about where this character lives around the world, what latitudes it lives at, and what is the treatment?
So this parasite normally live in the area close to the tropics. So warmer area, freshwater bodies is not able to survive chlorine water or salt water. So typical lakes, springs, rivers is when this amoeba can live. This is part of the environment. It's actually not a human pathogen. It's more like an accident. We are an accidental host for this parasite. So the parasite have different lifestyle stages in the life in the water. It's a flagellate. They move around when the nutritions or when the condition actually get really cold or difficult, they become a cyst and protect themselves for years. But if the condition actually is have a rich environment for nutrients, become an amoeba, and it's when it actually migrate. So when we jump in the water and the water rushed to the nose, when we felt like the water reached the brain, the amoeba sit in the mucosa in the nose and change to an amoeba. And then the amoeboidal shape start migrating and is able to access the brain to the floor of the skull. It's attracted to some neurotransmitters, but also the richness of the tissue make the amoeba to migrate. So what really causes the major problem is the immune response. So that's the major consequence. So controlling the immune response is one of the key parameters of the treatment. So be able to decrease the metabolism and some of the antiparasites that are currently available with a mix of some other antibiotics. So it's so few cases in the U.S. that there is no a full established. There is some protocol based on experience, but there is not a lot of true guidelines based on clinical trials because it's just around 140 cases since the 60s.
Wow. I remember seeing recently that the new Secretary for Health and Human Services in the United States, Mr. Kennedy, publicly said that he had had a brain-eating parasite. I need to check up on which it was that he said he had, but fascinating. Thank you for that. You've led efforts at AdventHealth where you work to implement next-generation sequencing, NGS technology, to identify mycobacteria, things like tuberculosis infections, more quickly and accurately. As a next gen specialist, can you expand on the emerging role of this technology and its power in infectious disease diagnostics?
So NGS is a powerful tool, but we still are looking for more clinical evidence and more guidelines to be able to establish as a permanent and more direct recommended diagnostic tool. Is in based on experience and publication, there is some clinical cases where direct sample NGS is probably better than what we currently have. In the case of endocarditis, where we have a lot of negative culture, that would be a great tool to if you don't have the organism identified previously. But there is another component of NGS that I think microbiology laboratory they want to get into the field of NGS and get experience is to be able to close some gaps. We have a good method of identifying bacteria right now, like the multitoph in the laboratory, the spectrometer, mass spectrometer. But for mycobacteria and fungi, we really don't have any new technology. So we still rely on microscopy and some biochemical. We rely a lot of in reference laboratory. So I think laboratories can bring this NGS to be able to identify this organism and close that gap, extend that to serotyping, genotyping, include this for more comprehensive antimicrobial resistant analysis, and molecular epidemiology. So I think this is the good step for laboratories to become comfortable with this technology and be able to then adopt a more widespread use of NGS for direct sample diagnostic. That I have based on my experience, I have the feeling that it's just a matter of time when NGS will become mainstream for many diagnostic infections.
It's amazing to think of the progress since the dim, dark days that I graduated from med school, the impact this had. And another groundbreaking piece of work you've been involved with your team is involving a Streptococcus pneumoniae serotype genotype service using whole genome sequencing. Can you tell us more about that technology and how it's going to transform the diagnosis and treatment of pneumococcal infection?
Yeah, so the pneumococcal infection is an infection that's being under control through vaccination, but still, especially in developing countries, we know that there is gaps in vaccination, and we have some serotyping circulating. Streptococcus pneumoniae is not really a common finding for us in children or adults when we compare with other pathogens, but still, I have a striking characteristic. A patient that have pneumococcal infection is the patient was vaccinated, was the Streptococcus have something that's called serotype escape. So that means the serotype that the patient have right now infection was not involved in COVID vaccine, or they have a vaccination breakthrough, right? The immune response didn't react to the vaccine and create immunity. So this is the two aspects of infection. So for us, that play a significant role to trying to understand and being able to manage treatment. We do susceptibility, we do identification. But what type of strain are we dealing with? So when we have a children with an invasive Streptococcus infection, the serotype play a role to define if we have a failure in vaccination or actually we have a failure on creating immune response. That can actually inform what is the next step after the treatment, if there is a boost or a change of vaccination. The other important aspect of that is the strain. So the sequence type is actually a look back, it's like a window to the back for the history of this organism. There is a strains that are known to be multi-drug resistant. There is a strain to be pan-susceptible, but have a high level of virulent factors, so make this organism a high virulent and spread. And then also the concept of serotyping switching. So when you have a specific strain that during time is able to switch the capsule and get multiple or different type of serotyping. That plays a big role because we can inform not only the vaccination status, but if we are dealing with an organism that have a significant history of being a significant pathogen or a highly virulent strain.
Interesting. And of course, the other element, which is the elephant in the room, is the so-called vaccine hesitancy.
Exactly.
I've never understood that term really. I, of course, come from the country that birthed the gentleman who's largely responsible for all this,
Mhm.
concern about the risks of vaccines. And the internet is an echo chamber, and once something has been said, people follow it through. Do you have any thoughts about the vaccine hesitancy?
It's interesting that the history is there. Streptococcus infection used to be a leading cause of mortality
Yep.
before antibiotics. Antibiotic came, Streptococcus were able to be controlled, but it still was a significant changes um, uh, but in medicine. The advances of vaccination create a level of the next level of protection, especially for children. I would be interesting to be able to talk a woman with a mom with 5 or 6 children from the 1900s, right, the beginning of 1900, and ask them if you have the opportunity to put an injection in every single one of your kids that will protect them from the infection that killed the baby of your neighbor, would you do it? So I think that will be an interesting perspective.
Yeah, that's the problem. God, I could go on forever about this. People have forgotten that diseases like measles, mumps,
Yeah.
tetanus, these killed, slaughtered millions of people, and just people have seemed to have forgotten about it. I know
Mm-hmm.
at the present in the United States there have been a number of measles outbreaks and
Right now we
mumps
have. Yep.
outbreaks. Yeah, it's shocking. Anyway, another huge one, which I mentioned in my intro, antimicrobial resistance, is growing global concern, something I'm very interested in. How can advanced diagnostics and data-driven decision-making aid in the classification of multi-drug resistant bacteria? And how can that influence treatment optimization and infection control?
So the perspective of antimicrobial resistance to is a little bit of like a work of intelligence over the organism. Be able to define the organism with criteria. What type of organism it is, what is the typical antimicrobial therapy, what type of resistance the organism already have, or a resistance that the organism can actually acquire. That define an organism in a better perspective. Sometimes calling an organism multi-drug resistant for some type of organism doesn't really mean a lot for the clinician. It just means that the organism is resistant, but doesn't give you idea of what type of antibiotic you should be expecting to be resistant. There is a better classification when we are defining the classification, something like ESBL, resistant to third generation cephalosporin, so you can use it for carbapenems. A CRE, a carbapenem resistant, a KPC organism. You know that there is some agents that have approval and actually indication for KPC, or an NDM, a major metallobenolactamase that is probably one of the most prevalent globally. So defining that give the laboratory a tool to be able to communicate with clinician meaningful clinical information to be able to treat the patient, not only to classify the organism as a label. And that also allows us to create cumulative data. If I have an organism with a KPC versus an NDM, how the profile of antimicrobial look for those two organisms, and how can I use this historical data to guide my next patient, right? To patient doesn't have to wait. Every hour a patient with sepsis, the mortality increase. So being able to put the patient in appropriate therapy is critical. The other aspect of that is, can we combine this phenotypic data with genetic data? What type of strain we're looking for? And this can actually enable an molecular epidemiology profile. We are able to know what type of organism we have, what type of resistance we can expect on them, what is the prevalence of those organisms. And if we find them, what is antibiotic that we normally are able to prove they have a clinical activity on those, and we can treat patients in advance. So antimicrobial resistance, um, not only from the testing perspective and treatment, but antimicrobial resistance can benefit a lot for data-driven information, because that can actually provide information ahead before any of the rapid testing or even susceptibility results are available.
Mm. So you mentioned KPC. You recently discovered a novel variant of Klebsiella pneumoniae, KPC 31, allowing you to identify its antimicrobial excuse me, susceptibility profile. What diagnostic tools enabled that discovery, and how was its resistance profile novel?
Yeah, so KPC31 has been circulating for a while. Has been found in Europe, has been found in the US. We found the first one in Florida. Um, the reason that this organism is significant is, even when it's a KPC, that we know there are carbapenem resistant mechanism. This organism behaves as an ESBL. That means it's not carbapenem resistant. So the problem with this phenotypic pattern is that many laboratories of this organism maybe is flying under the radar across multiple laboratories, because the method for identification that is carbapenem resistant is not the case with KPC31. So this was a patient with a sepsis. We were able to identify the KPC based on a molecular diagnostic. The phenotypic result didn't match the KPC pattern, so we actually were wondering what was the situation with this organism, what we're missing something, and we were able to do whole genome sequencing. At that point, we were able to identify the variant of this KPC as KPC31 that matched perfectly the profile of antimicrobial resistance. And this have a significant impact. Epidemiologically, we need to change the way that we screen this organism for KPC, because this organism is not going to be detected by a typical CRE screening. And second one, we need to make sure that we have patient and appropriate therapy, because some of the anti-KPC agents that we have currently on clinical use doesn't cover this organism. So I think create a significant gap in epidemiology. And if we see this is not the only variant, right? There is other variants for KPC2, and probably more will arrive. And this is just part of the selective pressure that we're doing in this organism with different treatment approaches.
Okay. Um, so many topics to discuss with you. I'm sorry that it's so truncated. I think this is just sort of warming us up for further conversations. There was a French Canadian microbiologist, Felix Derel, who I believe was credited with discovering the concept of bacteriophages, viruses to attack bacteria, if you will, to provide air cover for antibiotics. I know that FDA has approved them for animal and plant use, but not for human use. How far are we away from bacteriophages being used clinically for humans?
So bacteriophages is as you know, is nothing new in medicine, but it's something that modern medicine is actually embracing. Um, it's dealing with a living organism, have an opportunity that if the bacteria become resistant, the phage will overcome that resistance. So that's a fight that they have for billions of years. So they know very well how to do it. Um, there is some cases of compassionate use that phage therapy is being used successfully, and I think this is being driven the the the, the push for clinical trials. There is multiple clinical trials, some of them for UTI, for joint infection, ongoing right now. Hopefully presented to the FDA and get approval for some of the phage compounds or cocktails that could be available. I think the perspective is there. I think that it's a significant challenge because we're dealing with a biological agent, and there is a lot more to know. There is facilities and research across globally. But I've been lucky enough to collaborate with some of them where the phages is being evaluated by itself in combination with antimicrobial, the phage activity against certain organisms, the presence of biofilm, and how this can actually improve antibiotic therapy, not only using phage but in combination. I think that will be the future, antibiotic therapy and phage for certain entity, for certain organism. Um, and I think phage actually represent in infection disease treatment the better description of precision medicine.
Yeah, interesting. So watch this space, and maybe that's one of the answers for my next question, which is what emerging technologies for diagnosis or breakthroughs for treatment excite you the most, and over the next 5 to 10 years, where's your field heading?
So the the main one is NGS. I think NGS getting into mainstream and being able to provide the close the gap in diagnostic that we have right now with the different methods, especially with culture-based diagnostic method. I'm really excited to see NGS becoming more mainstream and become part of clinical guidelines for use, actually recommended. Um, the rapid susceptibility testing, I think we're seeing right now a switch on traditional antimicrobial susceptibility testing by broad microdilution. We are seeing a switch on rapid AST based more on metabolic analysis of the growth of the organism, or even the single cell visualization through microscopy. And I think that will make a change that probably will force ourselves to change the perspective of how we evaluate and measure that activity. Phage therapy is one of them, and maybe some even faster molecular diagnostics. Some of the PCR are becoming really fast right now, up around 15 minutes, 20 minutes. So I think that we're going to continue seeing that and be able to bring it closer to the patient for a rapid diagnostic.
And that sort of nicely takes me into my last question, sadly. If you were to come across a magic genie, well, you live near the Magic Kingdom, so if you came across a magic genie at Disneyland, a real one, I always get confused. Disneyland in Florida or Disney World? I forget which is which.
At Disney World.
Disney World. You come across a magical genie there, grants you three wishes in your area of healthcare. What would you ask for, Jose?
Well, first I will try to get one of those for a DB5.
No,
Um,
we're going to have to
no,
share it
no,
publicly.
okay. So I would think would be opportunities. I think I'm passionate what I do, and I'm in love with this field. I think that if I can have the opportunity, I will ask for the opportunity to be able to integrate all of these technology into a process that we can facilitate diagnostic or suspicion diagnostic to treatment in a more streamlined way. So I think for me that would be my first. Let me put together all this knowledge and all this technology to facilitate patient care. Second one is maybe a faster NGS, something that can be a push button. I'm pretty sure technology will. And third one, um, maybe a clear path for integrating artificial intelligence into the microbiology laboratory. I think that's one of the areas with the amount of data that we have. I think NGN can make a huge difference in diagnostic for what we have right
I think people sometimes forget, and we talked about the vaccine hesitancy space. I think people sometimes forget that if it wasn't for the work of people like you and your specialty, the impact you have had on human survival. I'm just a simple surgeon, and yeah, we've got better at what we do over the years, but the impact in infectious diseases has dramatically changed the survival of the human species. So, sadly, that's all we've got time for today. But I want to thank you, Dr. Jose Alexander, for being with us today. Real pleasure chatting with you, but also for everything that you and your specialty have done for the human race. And I'm sure you're going to continue to do a lot more. Thank you, sir.
now. I really appreciate the opportunity. It was a nice conversation. I hope we can have this conversation in the future, and maybe we can write together a DB5 in the future.
Well,
that's still my dream car.
Well, we'll talk a little bit more afterwards. I've got a couple of Aston Martin stories to tell you. So, to the folks listening to this episode, if you've enjoyed it, and I can't conceive why you wouldn't, please consider subscribing if you haven't already on Spotify or Apple Podcasts. Leave a review, say nice things, tell your friends. Check out the podcast. There's several hundred cool episodes on there. And join us next week for another fascinating episode. Until then, I'm your host, Dr. Jonathan Sackier. Thanks so much for being here. Please, everyone, stay safe, stay well, stay curious. Bye for now.